Hello, my name is Kayla Hannon. I'm a PhD student at Washington University, and I am investigating heterogeneity and late life depression. I am using a data-driven approach to determine biotypes within clinically homogenous subgroups to parse the heterogeneity in late life depression. Late life depression, which is when an individual 60 years or older has episodes of depression, is actually a major public health concern. It's highly prevalent. It's 13.3% of individuals in elderly life that have depression. It's associated with many negative health outcomes. All-cause mortality risk is increased. It is both associated with and known to increase frailty, stroke, cardiovascular disease, heart failure, Alzheimer's. So depression, especially late life depression literature, consistently shows small and inconsistent effect sizes in their results. And a very good paper that was recently published shows that as sample sizes increase, the effect size that you find in neuro-imaging gets smaller. And they argue in that paper that this is due to sampling variability, basically in a highly heterogeneous population. The actual effects that you will find will end up being very small, and when you have smaller groups, you will sort of spuriously find much larger effects. And this was sort of shown where even though previous smaller samples showed maybe higher but totally inconsistent results, a meta-analysis of larger sample size actually was not able to find a significant effect in late life depression, which they attributed to heterogeneity. So with all of that information, to me it seemed like it was important to try to parse heterogeneity in a large sample. And so that's what my project does. So I use the UK Biobank to investigate heterogeneity. Right now there are 50,000 older adults that have neuroimaging. And the UK Biobank is rich in both mental health measures and imaging measures. So I specifically use the recent depressive symptoms questionnaire, and for the imaging measures, I look at regions that have, in the literature, shown to be associated with LLD, and that includes frontal regions, basal ganglia, some parietal and temporal regions. And I look at both structural and functional aspects. So I look at functional networks, and then I look at gray matter volume, fractional anisotropy, cortical thickness, and also white matter hyperintensity, which is specific to late life depression. To understand how I'm going to parse heterogeneity, let's break down what levels of heterogeneity there are. So you can see here, this is the depressed population, highly heterogeneous. The DSM says that there are 9 symptoms for major depressive disorder, and an individual only needs to have 5 to count. So that means that individuals can have the same label, diagnosis, but have only one overlapping symptom. So first, we can parse based off of their clinical heterogeneity into sort of shared clinical profiles. But then there's the actual biology that's causing these symptoms. So you can further break down into their biological or neural heterogeneity. And so this is also how you can parse heterogeneity. So you can find individuals, isolate individuals that are high in one clinical feature, and find a group of individuals high in that one clinical feature, but then aren't high in any of the rest. So now they are clinically homogenous with no heterogeneity in their clinical features. But you can also break that down even further into their neural heterogeneity. So this is sort of why I'm looking for biotypes. Basically, there are possibly different biotypes that can cause each clinical presentation. And so I'll use clustering to determine and find potential biotypes. And so now some of these clinical groups, clinically homogenous groups, may not have further residual biological heterogeneity, and they may just have that one biotype. But some may have a couple. So what you see here is just an example of that. Some might break down further, and some might not. So here's an overview of my actual project. So these are the clinically homogeneous groups on top that I've parsed individuals into. I then did k-means clustering to determine different biotypes, and I did bootstrapping and cross-validation to determine stability of those biotypes, and then I also validated the biotypes based off of phenotypic measures that are important to depression but were not used in any previous part of the analysis to determine if they're significantly different as a sort of further step of validation. So the sources of clinical heterogeneity that I decided to focus on are the ones that the literature has shown to sort of be biologically relevant. So symptoms, I'll start with symptoms because I think those are the most clear and intuitive to understand. The symptoms that are shown to be important is anhedonia, which is like the lack of pleasure, inability to enjoy things you used to, that sort of classic depressed mood that we think of with depression. And then somatic disturbance, which is basically an increase or a decrease in motor activity. There's also types of severity. So acute severity would be when someone is actively currently high in symptoms, but chronic severity or chronicity is when you have like just many episodes in your lifetime, so you have sort of a high load. And then lastly, age of onset, specifically having late onset depression is shown to be important. So individuals could have no depression in their entire adult life until they got older. And so that's also shown to be biologically relevant. So here's a breakdown of how I actually categorize these groups. So individuals that were high, that scored high in anhedonia, were placed in the anhedonia group if they were then not high in any of the other things. The depressed mood group were high in depressed mood score, but low in all of the rest. The somatic disturbance group was high in their somatic disturbance, but low in all the rest. And then that just sort of keeps going down with the acute severity and late onset. And then there's also a heterogeneous group that is depressed but is heterogeneous, isn't high or low in any of these necessarily, and could have any of them. And this is just used as a comparison group. So I actually quantified the imaging measures using something called normative modeling, where you train on a population, a normal population, and then you test on the depressed individuals. So you get normative deviations, which are basically how individuals a measure of how they just differed from the normal population. So it's extra sensitive to biologically meaningful variability. So if we're thinking that the clinically homogeneous groups are in fact more homogeneous, then the idea would be is that they are more homogeneous and have larger effect sizes than the heterogeneous group, if heterogeneity really is causing those smaller effect sizes. So we actually tested that. I did an ANOVA to determine what the imaging measures that were significantly different between the groups are. And so here's just an example of one of the results. So you can see on the y-axis is the mean of the normative deviation for that imaging measure. And this is just the imaging measure fractional anisotropy in the corpus callosum, which is one of the significant measures I'm using as an example. And you can see here that the heterogeneous group, which is that like dark red color, has a lower effect size than all of the other groups, which does support what we were thinking. And then here is what they all look like. And you can see for pretty much all of them, the heterogeneous group is significantly smaller, or not significantly smaller, but is smaller than the homogeneous groups. So you can see some of the important things here. White matter hyperintensity is different in some of the groups. You see the hippocampus and the amygdala here. Okay, so now we want to parse the residual biological heterogeneity within those clinically homogeneous groups that you can see that we said we would do here. So this is functionally finding biotypes. So this is how I did it. I took the high dimensional imaging measures, the normative deviations, and then I reduced them down to a lower dimensional space to then do k-means. I repeated that process 1,000 times through bootstrapping to determine what the best number of clusters is using the maximum silhouette score. So the number of clusters that had the maximum silhouette score the most number of times in the bootstraps was considered sort of the optimal cluster solution. And then to determine the stability of those clusters, I did tenfold cross-validation and got their adjusted RAND index. So unfortunately, I only have preliminary results right now in the acute severity group. Hopefully by the time I actually present at OHBM, I'll have this a bit more fleshed out. But basically, what we found is that there were two stable clusters for the acute severity group that had an adjusted RAND index of 0.73, which is pretty stable. And then I did the validation only on the cognitive measures, unfortunately. The other ones are sort of in the pipeline. But we found that we looked at 4 cognitive measures, and one of them was significantly different between the two groups, which is an indication that these are stable and valid biotypes that differ between the groups. Yeah, to finish off, I want to thank my collaborators and coworkers and you for listening to my presentation. Thank you so much.